It's a pleasure to be here. It has been some time that I have been the last time at a roadshow. I think my first one was 2014. A lot of things happened, as you just saw in the presentation from Mattis and Steve. And it's really a pleasure for me to be here, especially. I started a collaboration, or we did a collaboration with our friends at Forge Biologics. And we present today together here on the stage. I will, as said before, introduce you the system in more detail, the features of the system and how it can help you in your separation task. And Frank?
Hi, I'm Frank Agbobo, Vice President for Process Development at Forge Biologics. Very happy to be here and to present on the case study we did. And so I'll be continuing the last half of the talk, talking through some of the data we generated from the single-use system.
So as it was alluded in the previous talk, we have a system here in the back, and everybody is invited to come after the session and take a closer look at the system. I have also a station where I can show you more the software. My background is I spent the last decade in developing innovative technology, but in a stainless steel, and I'm very excited that I joined the Repligen team, and at the same time, the Artisan team joined, and we became one, and I want to introduce you now to our platform we developed in the last 18 months. At this point, we have a quarter-inch system in the back, but we, as Mattis mentioned, we have system up to 1-inch tubing. So you can take the obverse columns from a 5 centimeter to an 80 centimeter or even bigger columns if you have self-packed columns. And you can cover the entire range, and you can scale up to these different sizes using our equipment. And Frank will show how the scale up can be done, and we have done it and show that it's reproducible. So let me start the presentation. This is a view, the same like mirror image, what you see in the back. And I want to highlight, go through the system, go kind of also a journey through the system and highlight the features, how they can accommodate your requirements. And so I will start the journey through the system at the inlet, and I'm going along the flow path. And just one point at the beginning, the flow path is a pre-assembled closed system. So assumingly we're starting with the aseptic connectors on the inlet valve block. We have 2 pumps on the system, so you can utilize the gradient formation, and each pump has 6 inlets. And if you remember the first slide from Steve, where he showed his first equipment, how crazy that was with single pinch valves, now we kind of compacted, put it very close, compact together in the system, in the Exo technology, and it's open here magically, and you can take a look inside. With the help of the overmolded technology, we don't need any barb connections there, so that further reduces the volume of the valve blocks. So we can have very close pinch points in the system and a very small hold-up volume. If you just think back on Steve's slide, what it took on space and volume, here this is 6 inlets, very compact together. So very low hold-up volumes there. And again, now you see the pinch points in the next animation, how close they are at the junction. So there's really 0 dead leg. So there's no chance of cross-contamination, that you get some buffers to interact with your feed solution. That is not possible. I want to point on one thing. We have an air sensor here on the inlet 1, so you can run your feed up to the inlet valve. And you can set this up with your recipe. So closing our valve, and we're coming now to the pump, and it's controlled via the flow meters. We have a clamp-on Sonitec flow meter, so there is no product contact of the flow meter with your product stream. And for the flow meter to work, we also use the Exo technology. So at this point, if you remember, this is on the discharge side of the pump. there's the highest pressure, so you can maintain your operation pressure of 4 bar at this point. And we have a pH probe there installed, so you can measure it and you can run it there at 4 bar. So going on in our process, we have the 2 pumps coming together. So with this optimized process flow stream, they're coming together in a mixing point. And again, there we're using this EXO technology, the 2 streams coming together. We have also a drain valve here, and then they're going to the next position in the flow path. But when you open the valve, again, very close pinch points of the valves, you could not accomplish that with single valve blocks. It's very close, very compact. There is no back mixing in here when the 2 streams coming together. It's coming together and forming the gradient at this point. Now we are moving to the next innovation. Again, there's another valve block for the filter, so you can bypass the filter or going through the filter. And as Steve alluded to it, we're using this filter, which is part of your flow kit, and we're using that also as a bubble trap. There is a level sensor on the filter. When the level in the filter housing drops below this level, automatically the vent valve will open, release the air, and you can use that filter housing not only to filter your stream, but also using it as a bubble trap to protect your column using this combination. Steve mentioned like a bubble trap at this size, 500 mils. We got rid of it. When you test the hold-up volume of this construct, of this flow path, I end up with 170 mils. So it's a significant reduction of the hold-up volume in your system, and this way you can really format a really good gradient, reproducible. Okay, we are going on. Just in case there is a bubble, oops, that was a little too quick. Oh, no, this doesn't. Okay. I had an animation, so you have to bear with me. There is on the outlet of the filter block, there is an additional bubble sensor. Just in case a bubble sneaks through that, you will detect it, and that allows you to do, through the recipe, some protection of your column. Now I'm moving through the flow path to the main intersection for your chromatographic process. That is a column valve. This valve enables you to bypass the column, to have an upflow and a downflow. Oops, that was one step too quick. I'm too quick here. I wanted to open this valve because you see there really nicely this innovation of the EXO technology, because we have here, okay, let it run. Sorry, it goes too fast, but you see there's 11 pinch points. So to reduce the zero dead leg in your flow path, so there's no cross-contamination, and it is very compact. Again, you can see it there firsthand in the back. We can open for you the valve, and you can be convinced by yourself, and you don't, with my animation. Anyways, when you come out of the intersection there, of course, we have post-column analytics. We have, it's again part of the flow kit. It's a closed flow kit, so it's a combination of pH and conductivity. The pH probe is installed in the instrument cup. So all the sensors are pre-calibrated, so you just have to pop them in. We're using dual wavelengths UV, 254, 280 for your gene therapy application, and we have also a standard offering of 280 to 300 nanometer for the UV. In case you want also the option, as you can see in the back, we have the pre-column analytics, pH and conductivity. And they are fully automated. They make decisions based on the readings there in your system. And just to show, I was talking about gradient performance. And I have done some experiments in the lab. So I run, you see here the red line is the flow. And so I run the 2 pumps together with a salt solution, and you can see how nicely aligned the conductivity is with the linear gradient. And then I step down, and you can see how quickly the conductivity is adjusted in the control strategy of the recipe. And that is important for your separation. Either you do an isocratic step gradient or a linear gradient, and just as a proof. Here you see then the outlet. Again, that's based on the EXO technology. Everything is very close together. So when you do your product collection, you don't have any losses in the tubing or any back mixing with your waste stream, for instance. Again, I just want to summarize at this point. So we have, for you, a closed flow kit, which includes all the components. So you don't need to calibrate your sensors. There are aseptic connection in between. Aseptic connections in between, where you have subflow kits you connect before you start. It's very easy to install. There is only one way you can install it. And again, come back there. And we have the combination of the filter and the bubble trap, which allows you to protect your column during operation. And in this way, how everything is optimized, you have an optimized gradient performance, and it's really reproducible. If you run multiple injection, multiple gradients, you will see that they are reproducible on your system. To go now, I have a few slides so you understand the importance of the hold-up volumes. Just stepping through quickly, like your normal, say, ion exchange process, where you start equilibrating your columns. You use inlet 2, equilibrate your column. You use the bubble trap filter combination, so protecting your column, and you equilibrate this. Now you're stepping to the load. You use the inlet 1, loading your column, and that could be based on time, volume, and it's all recipe driven. Or you can use the air sensor here on the inlet as a decision when you stop your loading process. And now when we step to the wash process, and it's important to see, say we're using the equal buffer, you're flushing everything out of the line onto the column. So there is no product loss. There's nothing lost. You put everything on the column. You can do your separation. You don't need to be afraid that there's any cross-contamination with any of the buffers. And then you can start with your elution. Again, you're using the 2-pump system. As I shown you earlier, it's very optimized flow path. And going through the system, you start your elution, your pre-column, your impurities coming out, pre-elution impurities come out initially that are compounds which are less retained on the column. And when you increase your buffer strength, your product comes out, and then post-product impurities, and you do then your regeneration step. Your collection can be done, again, it's recipe driven, based on UV signal, based on time, and you can make the decision. You configure your recipe accordingly. And so that would be the end. And I would like to mention, because I'm talking about recipe, I have a demo of the recipe editors there, so you can configure your process. So you're meeting your requirements, your GAM5 requirements, your 21 CFR Part 11 requirements. It's all part of our control strategy. At the end of your run, you get a batch report, which you can then put in your filing, and it's all meeting your requirements. And at this point, I would like to stop my presentation. If you have any question, please come at the end, and we can dive deeper into the system. And at this point, I give the mic to Frank and let him show the data. Very exciting work.
Thank you, Kathleen, for going through the system. And like I said, we've been doing a collaboration, working together with Repligen to test this new system. And right now, we have one in our lab, and we have a second one coming into our GMP suite. And so what I'm going to do is just go through this case study that we've done. So I'm going to start with a little bit of an overview of Forge Biologics, a company, what we do. And then I'll talk a little bit about our platform process. And then I'll go into the case study that we did, and then I will end with conclusions. So Forge Biologics, we develop gene therapy product, particularly AAV, and we are a contract manufacturing and development organization. And so if you are here and you're a small product developer for AAVs, you can talk to me afterwards because we offer end-to-end manufacturing and development services for AAV production. We also have our own therapeutic pipeline where we are developing our own products. So we understand the needs of people trying to develop these products. And so as a contract manufacturer, leverage that experience to be able to help our clients very well. So this is a picture of our facility. So this was taken during the winter. So you can see how it looks. And this is the front of the building right now. And again, I can invite you to come and take a look. And so we are building a 200,000 square foot facility. And when everything is built, we look to this being like the biggest manufacturer. So the unique services that we offer at Forge include end-to-end capabilities, like I was mentioning. A key component of this, especially being a multi-product site where we make different types of AAVs, producing different products. Single-use technology is a big part of this. And so you can see that we have a fully disposable single-use closed system. We have those for the upstream, and the Repligen system complements that to be able to fully have a fully single-use closed system. And so again, that went into how we did the collaboration and the work we've done. And also, we also have in-house analytical assays that is able to test performance. For cGMP production, we have capabilities from 50 liters to 1,000 liter run, also fully disposable single-use systems. And basically, we have our 5,000 bioreactors coming online later Q3 into Q4. We talked about our square foot, 200,000 square foot, and also mature supply chain. And it doesn't matter where you are in your stage, whether it's a gene with a dream, work with clients at every stage. And so if you're a client here doing AAV, we can talk after this. And like I mentioned, we have team members with lots of experience in regulatory support. And so we're able to help on that as well. In terms of our capabilities, we have clean room, single-use systems, segregated rooms, and also quality systems in place to be able to do GMP, as well as I mentioned the AAV and process development experience. So in developing a typical process, it involves a lot of steps. You have to develop your cell line, do media adaptation, like we all have to do, and then basically go through feeding studies, cell bank generation, and all the testing that goes with it, as well as then you do your small-scale evaluations, all those DOEs, optimizing your conditions. And so it's a lot of steps, going through clarification, affinity, formulation studies, and all of this take time, and it costs a lot as well to be able to do this. And so what we've done at Forge is to be able to develop a platform process that takes a lot of these steps out of the way. And so basically we have our Blaze, which is a research-grade production that we can quickly narrow this down to a few experiments that we run to show the process is working like we expect it to work, and then move it to cGMP production. And the value of being able to do this is the reduced timeline to be able to get your product to the market faster, and also the cost because of using the platform process. And so I'll go over this very quickly in terms of the process, just starting from a vial tour that you expand these cultures to the necessary volumes, and then basically go through your seed expansion, and then a 500 or 1,000 liter culture, and then from there you do your triple transfection, cell lysis, the standard depth filtration and clarification, and the affinity chromatography. And I'm going to be focusing a lot on this because that is what we did with the Repligen single-use system. In development, we work with, we have the small-scale bioreactors, the 1 to 10 liter range. We have the 50 liters. We also have a 500 liter in process development that we've been using for TOS productions as well. And then in GMP, we have from 50 to the 5,000. The case study that we did with Repligen was to compare the single use with a bench scale system. And so before we buy the system and integrate into our platform process, we want to be sure it's working well. And so the way we designed this was to do a 500 liter run, like any of the 500 liter runs I was talking about, and then take that through clarification, depth filtration, and then break it into 3. And so we have this material coming out, 3 1-liter material that was taken through a bench scale chrome system. And then we have 1 of a 50 liter taken through the Repligen system, and then 2 167 also taken through the Repligen system. So the chrome system is, first of all, preparing the system, and then equilibrating with a low salt buffer. And then we go through the loading step, a low salt wash, followed by a high salt wash, an elution, and then resin strip. And so same thing was done on the bench scale as well as on the Repligen system. So here I'm showing some data on the profiles that we were seeing. So you could see that basically during the wash and the elution, so we have similar profiles with the bench scale system. The only thing was when we got to the high salt wash, we were seeing this peak right here. And then when you look at the elution peaks, again, they are very similar. But then on the resin strip, we were seeing the smaller peaks over here, a little delayed compared to the bench scale system. And so this is another graph on the chromatograms that we pulled together. Again, you can see that we have the elution profiles for those 1 liter materials, and then taken through a 5 mil column. And then basically, this is the 56 liters of clarified lysate we loaded here, and then these 276s. And you can see again, the resin strips were smaller than what we were seeing with the lab scale system or bench scale system. The next thing we were looking into is also the yield. And so how much AAVs were you loading onto the Affinity, and how much did you recover at the end. And so, again, here you can see on the bench scale system, we were recovering about 60% of the material we loaded. And we were really impressed to see that on the quarter-inch Repligen system, we were getting 97%. I mean, that's close to 100% recovery on those that we did. And basically, you could look at the variation between the systems. You could see the variation is pretty similar in terms of the error bar from these triplicates. And so this is consistent with what we see at the bench scale as well as the large scale. And so basically, it shows some consistency in there. And also, the Repligen system, we've been able to show that we can do this up to 500. I'm not showing the 500-liter data here, but we've also tested it with 500-liter clarified lysate. And basically, we were able to get it to work there as well. So I will conclude by saying Forge has a platform process using single-use technologies by integrating the Repligen system. And this enables our clients to expedite their path to cGMP production, like I mentioned, savings on cost and time. And we saw similar elution profiles between the Repligen and the bench scale system. And we also saw a higher recovery from clarified lysate compared to the bench systems. And the single-use disposable system, this is integrated into Forge's platform. And so anyone looking to make AAVs, come to Forge to do your manufacturing. I would like to acknowledge a few key people to this. David Dismuke, our Chief Technology Officer, very instrumental in getting this going, as well as Ashley Craddock. She's our Director for Manufacturing. And so if you ever want to do manufacturing, you'll be interacting with her a lot. And then Donald Becher, one of my team members, another big contributor to this. And then Daniel and Steven, they've been really instrumental in the upstream, getting the bioreactors, and then Corbin and Ganesh on the downstream side. As well as Chris, he's worked a lot of long shifts with the team, with Kathleen and Rachel when we were doing this. And also on the Repligen side, it's been great collaboration. Just basically, it was long days when we were doing this. Sometimes show up around 6 o'clock to maybe 9 in the evening or late nights. But you can see it's been really good. So thank you, Kathleen. Thank you, Rachel, Rita, and David. And with that, I would like to also thank you for your time and attention. It's really a pleasure to be here, to be able to talk with you about this collaboration. And I look forward to any of you that is interested in talking about AAV production. Let's talk afterwards. Thank you.